Okay, the next portion of the chapter on cells is going to deal with transport processes. Now, in active transport, realize we have to draw on an energy source in order to move a substance against its concentration gradient, because this isn't something that would happen all by itself. We would say it's not spontaneous. So the two types of transport are primary and secondary active transport and vesicle transport. And this moves material that is polar or charged across the lipid bilayer, either into or out of the cell, using either ATP directly or indirectly in the form of a gradient set up by primary active transport. Active transport is going to require carrier proteins in order to function. A lot of these are integral membrane proteins that serve as pumps, moving material against their concentration gradient. In primary active transport, we use ATP directly to move material against its concentration gradient, and in secondary active transport, we use a gradient set up by primary active transport to drive material either into or out of the cell. In primary active transport, what we do is capture the energy from the hydrolysis of ATP in order to move material against its concentration gradient using an integral membrane protein as a pump. Examples include active transport of calcium, hydrogen, and the sodium-potassium ATPase, which is the most well-studied of the active transport pumps and uses 3/4 of the ATP in the body when it functions. It's located in the plasma membrane, and it is involved in both primary and secondary active transport of nutrients and ions. Sodium and potassium channels allow slow leakage down concentration gradients, which has to be offset by the sodium-potassium pump in order to maintain a property called membrane potential. And so this pumps against the pre-existing sodium and potassium gradients and maintains a high sodium concentration outside the cell and a low sodium concentration inside the cell. And the reverse is true for the potassium levels. The potassium levels inside the cell remain high and outside the cell remain low. This generates an electrochemical gradient that can be harnessed as energy for other types of transport. When we look at secondary active transport, we're talking about harnessing a pre-existing gradient in order to pull material in against its concentration gradient in order to allow the cell to get what it needs either from the outside or pitch what it doesn't want inside. An example is the sodium glucose symporter, which utilizes the gradient set up by the sodium potassium ATPase in order to drive glucose into the cell against its concentration gradient. So the way that this works is that the ATPase is kicking out 3 sodiums for every 2 potassiums it pumps into the cell for every ATP burned. That builds up or retains high sodium content outside the cell, and that is used to drive the movement of glucose into the cell. So when this transport protein opens up, the sodium is going to naturally want to flow down its concentration gradient. It will capture the glucose in addition to the sodium, and both will be deposited inside the cell. In an antiport system, 2 solutes would be moved in opposite directions. In a symport system, the 2 components are moved either into or out of the cell. So this is an example of a symport system of secondary active transport that moves material into the cell. Vesicle transport is also an example of active transport, and it requires membrane enclosed vesicles to move material around the cell. Okay, So in exocytosis, material is manufactured inside the cell, loaded in a membrane-enclosed vesicle, and using ATP, it walks along cytoskeletal elements until it reaches the plasma membrane, and then it will fuse with it and evert and eject its material into the exterior. Okay? In endocytosis, we have the reverse process. A portion of the plasma membrane will basically attach to something in the extracellular environment. It could be a solid, it could be liquid, and it may attach to it using receptors that are in the vesicle, or it may just be a non-specific engulfing of a piece of solid material. And then it'll pull it in, and again using ATP for the vesicle to form and for the vesicle to move, it will essentially bring that material inside the cell, where it could have a variety of fates. It might be fused with a lysosome. It might be utilized in transcytosis, where we move the material across the cell and exocytose it out of the other end of the cell, or it might fuse with another membranous organelle in order to drop off its payload. Okay? The trafficking of vesicles is an active process utilizing ATP to move material between organelles. Okay? So those are transport vesicles, whereas secretory vesicles are there to excrete material manufactured inside the cell, and endocytotic vesicles and phagosomes are used to bring material into the cell. You can see here endocytosis and transcytosis, right? You bring material in. Protein-coated vesicles are one way we do this. This is a receptor-mediated example in which the solid attaches to receptor proteins embedded in the membrane. The membrane invaginates in this region. That is an ATP-dependent process. And then cytoskeletal elements in ATP move that vesicle to perhaps another membranous organelle, or perhaps to the other side of the cell where it exocytoses. Okay? So it all depends on the fate of what it is we're bringing into the cell. In phagocytosis, pseudopods, which that is a term meaning fake feet, engulf the solids and bring them into the cell interior, forming a vesicle called a phagosome. This is used by the macrophages and some white blood cells. It also promotes movement by amoeboid motion as a precursor to engulfing the particle. The cytoplasm flows into these temporary extensions, and it basically creeps around the object and then pulls it in. And again, all of this is ATP dependent. Okay? In penocytosis, what we're doing is we're bringing in material such as a liquid in order to sample it, right? So what happens is that we invaginate a portion of the membrane, we pull the liquid inside. The nutrient absorption in the small intestine is one of the places where this happens, and the membrane components end up being recycled back to the plasma membrane surface. In receptor-mediated endocytosis, we have specificity between what we're bringing in and the receptor that it binds to, which is going to be an integral membrane protein. Cells use this to concentrate materials that are in limited supply in the external environment. Clathrin-coated pits provide the main route for endo and transcytosis. Uptake of enzymes and low-density lipoproteins, as well as iron, insulin, and viruses, as well as certain toxins such as diphtheria toxin and cholera toxin, all move into the cell using this method. Coat proteins include caveoli, which capture specific molecules such as folate and tetanus toxin and utilize transcytosis to move across the cell. They can be involved in cell signaling, but the exact function is unknown. The codomer is what's going to function in vesicular trafficking, moving the material from one location in the cell to the other. In exocytosis, we have activation by a cell surface signal or a change in membrane voltage, a substance gets enclosed in a secretory vesicle. Vesicle snare proteins on the vesicle attach to target snare proteins on the plasma membrane. They bind and fuse to each other, and this is functioning in hormone secretion, neurotransmitter release, mucus secretion, and ejection of wastes from the cell. We talked earlier about the fact that the membrane potential is a type of stored energy in the plasma membrane that can be used as a source of power to promote activities such as nerve signals and muscle contraction and transport in a lot of different cell types. The resting membrane potential indicates that the inside of the cell is negative relative to the outside, and this is due to the differential permeability of sodium and potassium. The membrane is much more permeable to potassium than it is to sodium, and the fact that the sodium-potassium pump is always kicking out more positive charge into the exterior than positive charge into the interior, and the fact that large negatively charged molecules such as proteins are locked inside the cell cytoplasm and they cannot escape. The range of this negative membrane potential is from between minus 50 to minus 100 millivolts, depending on the cell type, and it's used as a source of power, just like a charged battery, to move material from one location to the other. Okay, in the sequence of events here, potassium tends to diffuse out of the cell, while sodium tends to diffuse in. More potassium diffuses out than sodium diffuses in because the membrane is more leaky to potassium than it is to sodium. So this results in increased loss of positive charge from inside the cell. Okay, the sodium-potassium pump is always working in the background, and those negatively charged large anions are locked inside the cytoplasm and cannot escape. And all of that contributes to a resting membrane potential that in many cells is around minus 70 millivolts. Okay? Normally chloride is not a player here. It doesn't influence resting membrane potential. The concentration and electrical gradients are basically exactly balanced. And this resting membrane potential actually requires a lot of energy in order to maintain. The sodium-potassium pump is the main player here, right? It continuously ejects 3 sodiums from the cell, carrying 2 potassiums in. A steady state is maintained because the rate of active transport is equal to and dependent on the rate of sodium diffusion into the cell. Neurons and muscle cells, which are excitable cells, change this resting membrane potential by opening voltage or ligand-gated sodium and potassium channels, and that results in an electrical signal called a depolarizing wave that can propagate through the cell and serve as a signal for the cell to do something. Cells can interact directly or indirectly, responding to extracellular chemicals. This always involves glycocalyx proteins, which include cell adhesion molecules and plasma membrane receptors, as well as voltage-gated channel proteins. The CAMs number thousands on about every cell of the body, and they anchor to the ECM or to neighboring cells. They aid in the movement of cells past each other and attract white cells to injured or infected areas, sort of like a signal flare. They stimulate synthesis or breakdown of membrane junctions, and that controls the permeability of tissue layers. And they transmit intracellular signals in order to direct cell migration, proliferation, and specialization. In contact signaling, touching and recognition of cells has an effect on growth, right? When the cells grow into what we call a monolayer in many tissue types, then further growth in the vertical direction stops, okay? And this is something called contact inhibition. Cancer cells break this rule and tend to pile up in a monolayer or within a tissue. And that's because they've sustained a mutation that changes the way in which they regulate their cell cycle. Chemical signaling is the interaction between receptors and ligands that changes the activity of the cell. What happens is that when the ligand and the receptor attach to each other, they undergo a shape change, and that shape change is then relayed inside the cell to produce a response that makes it more likely that the organism will survive, okay? The type of response depends on the ligand and the receptor that are involved in the hookup. You can see an example of this in what we call G proteins, okay? In a G protein, what we see is something known as a signal cascade. The ligand binds an integral membrane protein, causing it to change shape. In the case of the G protein at that point will be induced to kick out a bound GDP and bind a GTP, which is an energy molecule. It'll disengage from the G protein, shuttle across to another integral membrane protein, which can then manufacture a second messenger. Frequently, the second messenger is something called cyclic AMP, which is made from ATP. The result of the second messenger is to hit downstream protein targets that hit more downstream protein targets, and eventually the cell responds. The reason for such a complicated way of relaying signals is that when you generate what we call a signal cascade, what you're doing, in fact, is you're magnifying the signal at each step so that it's not a one-to-one correspondence here. For instance, every single, let's use the laser pointer here, for every single receptor and ligand interaction that we see, multiple G proteins can be activated, which can activate multiple downstream membrane targets, which activate multiple, let's say, cyclic AMPs, which activate multiple downstream targets. And so the strength of the signal grows with each step, and the speed of the signal and the response is quite efficiently magnified. And this is something that we see not just inside cells, but we see cascades outside cells as well for the same reason, in order to magnify the signal and make it more effective, okay? This is generally the case when the signaling ligand is charged or polar, okay? If the signaling ligand is nonpolar, frequently the receptors are inside the cell and the complex has to go to the nucleus where transcription is activated and proteins are then manufactured in translation, and that results in the cellular response, and that usually takes more time. And that's why when we're talking about a polar or a charged ligand, the response time is much less than if we're talking about a nonpolar ligand. Okay? So, that brings to an end this portion of the discussion. I will see everybody in the next installment.